Hello guys, I'm going to be doing dialysis and diffusion today. But first, I'm sorry, osmosis. But we use dialysis bag. This is dialysis bag. I'm going to cut about 12 centimeter, just like that. Okay, I call 12 centimeter. And then from one end, I put a string, tied it up very well. Tie up the knot right here at one end, and then as your lab manual said, of course you have your lab partner and make a couple of knots, and then here's a string. I cut up another string to be ready for the other. I just want to show you how it's done. So cut up the other string, and then you measure 10 ml of distilled water. I just put distilled water in here, about 10 ml, and then you measure it right here. And then of course you have to do sugar solution. Where is my sugar? 10% glucose. You have to do that. And then you have to make the paper very, very wet. So I'm gonna stick it in here, make it, or you can put it under the sink and make it very, very wet. And it's just like grocery bag. It comes up open, I hope, like this. So now you have a bag. It's a tube. Both ends were open. That's why I closed one end. And then you pour. I hope I don't spill any. I might, but that's okay. This is for demonstration purposes. Okay, of course it's difficult. You can get it in there. You can use your imagination and accuracy, and then put the rest of it in here. Eventually, this is gonna be, I'm not gonna put all of it, waste your time. And then you put the other string at the other end, and then you put it in a, I hope you guys can see this. I don't have, as I said, I do not have a cameraman. It would be nice to have a cameraman and do this. This is Corona time. We are trying to reduce as many bodies as here. So you have a bag full of fluid in here. You put it on a weighing boat before your 30 minutes, and you go and weigh it on the scale, which I showed to you guys for the metric system, the very beginning of semester, how to use the weighing boat. So you go over and weigh it. And then right here, I put the bags. Oops, I hope I don't tip over anything that I'm not supposed to tip over. Right here, I'll try to show you guys. So bag A has sugar in here. I put 10%. I already put 10% sugar in the bag and water in the beaker. Okay, and then bag B, water in the bag and water in the beaker, and and C, it is water in the bag and sugar in the beaker. I waited put them in a weighing boat every 15 minutes. I went to the scale and weigh them. So the way they look like now after 30 minutes, let's look at them. Bag A, what happened? The water went from the beaker into the bag, so the weight of the bag increased. Bag B, if you will look at it, it stayed the same. Not much happened there, because inside of the bag and outside of the bag, if you want to compare them right here, this is a little bit heavier. We can go to the scale and weigh them. And bag C, definitely, as you can see, lost a lot of water. Look at bag C, because there was water in here and there was sugar in the beaker, and the water molecules left the bag, so the bag shrunk and became that size. So that's pretty much an example of osmosis. This is what's happening in here, osmosis. One more example of osmosis before I go on and talk about anything else. This is alodia leaf right here. right here is alodia leaf, and I put one leaflet in there. Now I'm going to add some salt solution to it, a little bit on the side of the slide. It would be nice scientifically add like, okay, now it diffused throughout the slide. Let's go look at them under microscope right here. Oh, I did not adjust it. I am uploading other stuff. So let me grab the minimize. Let's look at this. Okay, so this is the one that I have not. These are chloroplast. I hope. Let's see if I can. You guys can see it or not. Yeah, I hope. Yeah, very good. Okay, so these little dots. These are dots. You can see here. These are chloroplast. This is cell wall right here. Another cell wall. So you can see plant cells are rectangular. Right here, you can see them. They're rectangular, and the chloroplast are right here, the green dots, and they're moving around. That's called cytoplasmic stream. Now, let's take that off, and I'm gonna go back. You remember the microscope, and let's put the one that we added salt to it. Okay, I'm gonna add the one with the salt. So this is. Can you guys see this? This is on the 4x. Okay, so I want to magnify it and go to 10x. Of course, plant cells have cell membrane and cell wall, and then this is the 40x. And as you can see already, some of these, I hope you can see them very well. Maybe I should increase the light just tatty. I can see it on the microscope, and then, yeah, great. I hope you guys can see it too. So you are seeing, I'm seeing already, the cells are shrinking. This is shrinking, and what is left is cell wall. And if I wait a little bit longer, you will see the cells shrink. And so the cells are shrinking when we added the salt solution. What caused this shrinkage of the cell, because the environment was salt, the water from inside of the cell went outside, so the cell shrunk. Okay, let's look at the end. We'll come back and look at this again after a few minutes. This is different than the other one. Then the other experiment that we're supposed to do for this lab is diffusion. I already put a few of these for you guys. I added, if you would, I added Congo red or red dye. These are food colorings right here to a beaker about half an hour ago. And I added methylene blue, which is solid. You can look at it. It's a powder. And I added to the beaker and the gel. This is the agarose gel, semi-solid. If you would, I gotta make sure you guys can see this a little bit more down. Okay, now it's good. Okay, so already done this, and then I'm gonna do it again. So you can see this, it's already diffusing. It is diffusing, there is no membrane involved in here. So I'm gonna grab 2 more plates, this one and this one. Just do it right now so you can see the difference between the two. So here is food coloring, doesn't matter. Right here, oh sorry. Oh, sorry, okay. You see what happened? Right now, in a few minutes, it's gonna become like that. I get everything stained at this school. Sorry about that. Okay, and then the methylene blue right here. I grab a tweezers. I do not want to get my just the tiny patty right here. if you look at them, of course you saw it in a few minutes, hours, it becomes like that. So that is diffusion of solid in semi-solid, diffusion of solid in liquid. Okay, this is diffusion of liquid food coloring in semi-solid, and of course diffusion of liquid in liquid. So let's do that again. Right here, I'm gonna put this in the front so you guys, I have to check, make sure you guys are seeing that. Yep, you can see it, very good. So right here, I'm gonna put the Congo red, same thing. And leave it on the bottom. Okay, right there. I hope you can see it's going to diffuse throughout. You guys see that? You have not diffused it yet completely. Okay, it will diffuse, and this one was done half an hour ago. Look. Oh, where are you? I had it. Here we go. Okay, so that was liquid in liquid. This is the diffusion of solid right there in liquid. Of course, it takes some time to be dissolved like the other one. So there are 4 diffusions that I'm demonstrating here for you. Solid in semi-solid, solid in liquid, that's 2. Liquid in semi-solid, and liquid in liquid. So there are 4 different type of diffusion, and I hope you would know them. And I'll take a picture and I'll put them in your lab in the modules. All right, let's look at this. Perfect. You can see this now. It is perfect. I can see it perfectly. I hope you guys can see it too. I wish. Yep, this is great. Right here, you guys are seeing this one, this circle right here. All of the water left the cell, so the cell became, of course they died, the cells are dying. You can see them here, they're all over. You can see it a little bit better than the other ones. All right, so I'm done with this. I tried to take a picture of these and put them in your modules.